Well, hello. Welcome to our second of our webinar series for this academic year. My name is Shubha Ghosh, and I'm the Crandall Melvin Professor of Law here at Syracuse University College of Law, and also the Director of the Technology Commercialization Law Center. And on behalf of my colleagues and the New York State Science and Technology Law Center, I'm really pleased to introduce Professor Jacob Sherkow, who directs the Innovation Center for Law and Technology and is an associate professor at New York Law School. He's going to talk about CRISPR patents and ethics, law, and social impact review. That's what LC stands for, and I think I wrote it down this time, so I remember. So welcome, Jacob. Thank you for coming.
Thank you.
Look forward to our interactions.
Thank you very much for having me.
And for those who are listening and tuning in, please do send your questions as we progress. Professor Sherkow will take us through some of the battles surrounding CRISPR, and I will be interrupting occasionally with questions, and I think we'll have a fruitful discussion until about 2 p.m. So it's all yours.
Thank you very much, Shubha. Thank you very much for having me. I am here to talk about the CRISPR patent estate and some of the ethical, legal, and social issues that are arising from it. So as many of you who are tuning in know, there recently, in 2012, was elucidated one of the most important gene editing technologies that we have seen today. The principal participants obtained a number of patents covering foundational aspects of that technology, and that ownership of intellectual property over the foundational aspects of this really important technology has raised a number of issues that I and a number of other scholars in this area have examined recently, and it's those that I'd like to talk to you about today. So if we were to think about the structure of my talk for a moment, I'm going to talk a little bit about CRISPR, what the technology is, and talk a little bit about the patent dispute that is currently playing out between a number of the principal investigators. I'm going to speak to you about some of the issues that arise when we mix patents covering foundational aspects of technology with research universities, especially with respect to profit and collaboration, in other words, scientific research collaboration, both within and across institutions. I'm also going to talk about some of the difficulties that arise from the entities that do sub-licensing from some of this technology, what George Contreras at Utah and I call surrogate licensors. And then lastly, I'm going to talk about some of the ethical gatekeeping norms for the CRISPR patent dispute. That is, ways in which the patents covering foundational aspects of the technology may actually promote rather than hinder their ethical use in the research marketplace. So let's talk a little bit about CRISPR, the technology, generally speaking. Since Watson and Crick here first discovered the molecular structure of DNA back April 25th, 1953, it has been the holy grail of molecular code. And by the genetic code, I mean the A, C's, T's, and G's that make up your genome, much as you likely already know, right? There have been some attempts to do that from 1953 until now, but for a number of reasons. They have been either imprecise or expensive to use or simply ineffective, right? CRISPR seemed to solve all of these at one shot. The canonical view of how CRISPR, the technology, gets developed occurs back in August of 2012 with the discovery of CRISPR as an engineerable genetic editing system. The discovery was memorialized in a paper by Jennifer Doudna on the right here, she at University of California, Berkeley, and by Emmanuel Charpentier, then at University Umeå in Sweden, now the director of infectious disease research at Max Planck Institute in Germany. They published a paper in 2012 in the journal Science describing how we could use the CRISPR system mediated through one particular strand of RNA, that's a cousin of DNA, to effectuate genetic editing in bacterial cells and in test tubes, in other words, in vitro, as researchers would have liked. Right? So the canonical story goes, Feng Zhang, a researcher at MIT Broad Institute at Harvard, at the at the Broad Institute of MIT and Harvard, reads Jennifer Doudna's and Emmanuel Charpentier's paper and figures out a way to modify this system to enable it to be used in eukaryotic cells, that is, the cells of higher organisms, like humans, for example. In doing so, Feng Zhang was able to move CRISPR as a technology from arguably one of the most important research tools to molecular biology since PCR in 1985, from that into the potential for an actual gene-editing human therapy. That is at least how the canonical story goes, but there often is a person missing from this equation. That person is Virginijus Šikšnys at Vilnius University of Lithuania. Virginijus Šikšnys was actually the first to realize the potential of using CRISPR as an engineerable system, but because probably he was at Vilnius University in Lithuania as opposed to a major research institution here in the United States, he had some difficulty getting his paper published. So although it appeared that he was the first scientist to recognize the potential for this system in the way that we have now come to expect it to work, he was not the first to publish it. Nonetheless, at the same time, all of these researchers involved, Virginijus Šikšnys, Jennifer Doudna, Emmanuel Charpentier, and Feng Zhang, all obtained or at least filed for patent applications on their particular inventions when they discovered the technology that they were researching. Virginijus Šikšnys, believe it or not, was actually the first to file for a patent application covering an engineerable form of CRISPR. That was back in March of 2012, although his patent did not issue until May of this year, May 2017. Jennifer Doudna and Emmanuel Charpentier filed for their patent application in May of 2012, just shy of 2 months after Virginijus Šikšnys here. And Feng Zhang files for his first patent application in October of 2012. Feng Zhang, however, or Feng Zhang's attorneys, decided to fast-track Feng Zhang's application through the PTO and also limit that application simply to using CRISPR as a human therapy, or using CRISPR, I should say, in eukaryotic cells. So ultimately, where does that leave us today, September 2017? That leaves us in a situation in which Feng Zhang's fast-tracked applications covering eukaryotic applications of CRISPR have issued. Virginijus Šikšnys' fundamental application for using an engineerable form of CRISPR has issued, but Jennifer Doudna and Emmanuel Charpentier's application is still pending at the PTO after losing a particularly contentious interference dispute with Feng Zhang. Right? That is the current situation that we have today. All of the patents in this particular area, we consider those to be the foundational patents that cover the CRISPR technology. Using iterations of CRISPR going forward, using it as a particular therapy, using it to do research in particular types of cells. In theory, at the very least, one would need to obtain a patent license from all of these 4 major players here to practice that technology going forward. So it is this reason why the scientific dispute, and understanding who came first and when they filed for their patent applications, is particularly important to understanding what the landscape is for foundational intellectual property covering this technology.
So Jake, sorry to interrupt, and for our audience out there who may have just joined us, I notice a number of people have joined us, please submit your questions by typing them in, and they're anonymous. I'll just read the questions. But I did have a question about just the Doudna application. Where exactly do you think it's likely to issue? What exactly are the obstacles for that at this point, given the other 2 have issued and the defeat with the interference?
Yeah, so University of California, Berkeley and Emmanuel Charpentier lost their particular interference dispute against Feng Zhang. The PTAB, that is the appellate board within the United States Patent and Trademark Office who conducted the interference proceeding, ruled that there was not, in fact, an interference between Jennifer Doudna and Emmanuel Charpentier's application and Feng Zhang's patents. That what Feng Zhang did was actually a new invention and was different, and therefore that they decided to, I don't know if this is the appropriate word, rescind the interference that was previously declared. That decision is currently being appealed to the U.S. With that being said, the standard of review for reviewing interferences at the Federal Circuit is particularly high. For the Federal Circuit, they would need to determine that the PTAB made its decision against substantial evidence, so that there was not enough substantial evidence to declare that there was not that there was, quote, no interference in fact between Zhang's patents and between Doudna and Charpentier's patent application. That certainly does not seem to be the case here, where the PTAB reviewed expert reports from at least 3 witnesses, conducted a public hearing, took in hundreds, if not thousands, of pages of written argument and testimony from scientists and from lawyers. It seems as if anything satisfies the substantial evidence standard, this is a case that probably meets it.
So if there is a reversal by the Federal Circuit, which you're suggesting is unlikely, would it go back for a continued? I mean, they could reverse to say that the data application did have a feng shui patent. Would that be invalidated?
Yeah. So there was actually no evidence presented on priority yet. That by declaring no interference, in fact, at this early portion of the interference, in what interference attorneys call the interlocutory phase, there wasn't any evidence, or at least there wasn't enough evidence taken with respect to priority. So to the extent that the Federal Circuit reverses here, they're almost certainly going to have to kick it back to the PTAB to then actually to conduct a full-blown trial on things like priority and scope. Right.
And if there is an affirmance, then the data application would just continue in the prosecution.
That's right. Although now we're left with the situation where Virginia Sherkowsept has his earlier filed patent, which now becomes a 102E problem for Doudna and Charpentier. 102E says that a patent that issues after an instant application that was filed before it may be an anticipatory reference, right? So now we have this Sherkowsept patent that has been issued that could be a 102E problem for Doudna and Charpentier. And we have Feng Zhang's patent on the other side, which if the Federal Circuit affirms means that eukaryotic applications are different from what Doudna and Charpentier have. I don't know how much scope that leaves Doudna and Charpentier to carve out in the middle, even if they do ultimately end up winning at the Federal Circuit.
So I don't want to linger too much on it. You have a lot to cover in this way, but it's probably worth on this point.
Sure, it's great.
Here, but I just want, as I understand,
and I'm also happy to take any questions that people have while I'm speaking. If you would like to interrupt me, please feel free.
I just wanted to interrupt here just because it's kind of at this probably the appropriate question to ask. It's interlocutory.
Sure. That's right, exactly.
So. But the Doudna application has been granted by the European Patent Office, is that correct?
Yeah, so a patent on generally the same specification. The European Patent Office heard a bunch of arguments in the kind of pre-issuance phase. It's not technically called an opposition proceeding. It's called something else. If people keeping score at home know it's that called, please, write in. So the European Patent Office, with respect to that, has sided with Doudna and Charpentier over Zhang. It appears that the Chinese Patent Office has also done the same. So that's the kind of landscape that we have now.
So here's a big question that's building up with all this background. Not a big question.
No,
this is going to be not disappointing to people when it's not a big question. But so the reason we're doing the interference is that it's under the pre-AI American Invents Act, which is a first to invent system.
Yeah.
And the European system, I think, is a first to file or some variation on that. So if this dispute was coming up under the AIA, which is in the U.S., which is a first inventor to file system, would the result have been more like in the European Patent Office, you think? Or, I mean, I know this is really speculative, but this is really getting to the comparative of the system,
So one of the interesting things with respect to the interference is that I don't think there's much dispute concerning priority, right? Nobody has claimed, or at least no one from Zhang's camp has claimed, with some apocrypha that's floating out there that I don't want to ascribe to anybody, right? No one has claimed that Zhang actually came up with a single guided RNA engineerable form of CRISPR prior to Doudna. It seems to be whether or not people in the art recognized that it could be used for eukaryotic applications and what things needed to be done to use it for eukaryotic applications going forward. So it doesn't seem, or it is, I think, affirmatively not the case that any of Zhang's references are used as prior art against Doudna now, even under a first-to-invent system. So to the extent that we are under a first-to-file system, I assume that that would come out similarly.
Interesting.
I think the big difference, and I've actually written about this in a recent article in EMBO Reports. EMBO is the European Molecular Biology Organization. They come out with a really wonderful journal once a month that, aside from publishing really interesting research from European molecular biologists, also publishes interesting perspectives on things like European patent law. I actually wrote a perspective here that suggested that one of the real differences between how things come out in the United States and how things come out in Europe with respect to non-obviousness is the United States focus, the USPTO's focus on whether there was a reasonable expectation of success to drive a genus of a technology toward a particular species. That was the crux of the decision with respect to the interference at the U.S. Patent and Trademark Office. That's just not something that the European Patent Office looks as closely to. There seems to be a better alignment at the European Patent Office with how scientists view what a reasonable expectation of success is, or how scientists view the adaptation of a technology for further purposes. There seems to be better alignment of science and the law in the European Patent Office than maybe there does in the United States.
Interesting. So we do have a question from an audience member, and I think
it's Christy. Hi there, Christy.
Okay. Try to be anonymous here, Chris.
Oh, sorry. Okay. Sorry.
It may or may not be this person named
Okay.
Christy.
All right. Good. Fair enough.
Let me just read the question.
Sure.
So, and I think you sort of answered it. How is Doudna successful in Europe over Zhang? What argument or legal mechanism worked there in her favor?
Yeah. So again, the kind of crux of the interference dispute in the United States was whether or not someone looking at Doudna's contribution to the invention would have had a reasonable expectation of success in getting the technology to work in eukaryotic cells. The differences, the legal differences in assessing that questions in Europe versus the United States seems to have driven the difference in the decision between the two places. That's what it seems like.
That's interesting. Great.
And I didn't realize that questions were anonymous,
I'm just joking.
I'm sorry.
I'm sure we're not liable for anything.
Yeah. Okay, good. All right. So happy to continue with the discussion of some more of the ethical, legal, and social.
I just, kind of wanted to...
No, please. So just a brief review, since we've talked about all this with the technology and how the technology actually works. Okay. This is the by Doudna and Charpentier in a review article in Nature of their invention. This is a canonical description of how the technology works. Note the publishing date is 2013. That is after their important article comes out in Science, but also after that they note that Zhang is working on this in eukaryotic cells. Hence the two, I don't know if you guys can see my mouse here, the two eukaryotic applications at the bottom of this graphic. But still, what we have is we have this protein called Cas9. This is an enzyme which we call a nuclease. It works essentially by cutting DNA. The only thing to activate Cas9, this is what makes the CRISPR system the genius system that it is that purple molecule that is attached to it or inside of it that you see here. Jennifer Doudna and Emmanuel Charpentier were responsible for taking this, essentially understanding that this single purple strand was the thing that did the work. This single guide RNA was the thing that both directed the enzyme to the particular portion in the genome that you wanted it to cut and also activated such that it did the cutting, right? After the genomic DNA is cut, it can be repaired in a number of different ways, including being repaired with the green donor DNA that you see there, which could be the DNA of your choice. You want to insert a gene into a particular portion of the genome, you can. You want to remove a particular area of a gene in the genome. In theory, you could do that too. There's a virtually limitless number of modifications that you can use this system for, given how precise it is. That is how CRISPR works. Right?
And can I just ask, this is just a naive technical question,
sure.
But I'm just curious about this for, eventually down the road they talk about CRISPR kits you have at home. That's decades away probably. But here you're physically altering as a way of physically cutting and altering the DNA. If you wanted to do some projection, so how does this relate to, for example, proteomics or bioinformatic research where you take the data and then you want to see what your, three-legged zebrafish.
Oh yeah,
take.
oh
So
yeah.
could you explain that a little bit?
Sure,
Yeah.
absolutely. So one of the really amazing applications of CRISPR is its use as a screen. That is essentially taking a bunch of randomly developed or randomly selected pieces of this purple single guide RNA, taking thousands or tens of thousands of them. You could screen a genome, or you could screen a group of cells to see what each individual one of them does, right? To see what each gene does, right? Researchers are using this right now. Maybe the lead researcher in the United States who's using this right now is Neville Sanjana at the New York Genome Institute and NYU. One of the things that he is using CRISPR screens for that is likely to be of interest to many people is to try to uncover the molecular basis for autism. There's a number of neuronal cell autism models, and that by using tens of thousands of different genes all effectuated through this particular guide RNA here, we could do a good job at not just silencing them, not just turning these particular genes off, but actually removing them from the genome and seeing what effect they have, if any, on neuronal development in this model that we have. There's a number of other different ways to use this too, but that is one particular area that CRISPR aligns with some of the more recently developed bioinformatics research that we have out there now.
Yeah, so the patents at issue in this case are process patents, basically, right? Or are they?
Mainly, they are methods of using. The method of editing a eukaryotic cell comprising so on and so forth.
Yeah. So let's just delve into that, just on the IP implications for this, for those who use the CRISPR technology. So let's say you do have the edited genome. Do you think that could get patent protection? There's really a potential problem here for patent thickets, right, where everybody's doing different edits, and
yeah, I
assume there'll also be maybe some copyright protection to the extent we're thinking about these genomes as databases, or.
I guess that's possible. Okay. I guess one interesting thing to think about is the databases that are used for this CRISPR screens, right? Whether or not the database itself would receive copyright protection, but to be clear, that doesn't mean that the actual reagents, that is the actual single guide RNA molecules, are going to have anything to do with the Copyright Act, right? So don't know exactly what the enforcement capacity of protecting something like that is going to be. With respect to the actual, edited genomes being protected, I think ultimately the answer is probably no. I think that the answer is probably no, both because DNA or kind of genomic DNA that has been only routinely modified is probably no longer patentable after the myriad genetics case, right? Okay, DNA is no longer patentable just because it has been isolated and purified from its surrounding genome. Well, I guess the flip side of that is that a genome is no longer patentable just because there's something that's been isolated and purified from it, right? I think that that's probably true. Furthermore, to the extent that you wanted to patent some of the reagents that are being used here, you wanted to patent some of the single guide RNAs, and I think the Myriad 2 case is instructive. The Myriad 2 case is the case in which after the Myriad Supreme Court decision case, Myriad Genetics still had patents covering a lot of what they called cDNA, primers and probes. They're snippets of DNA, just like our purple guide RNA that we have here. There, the District Court of Utah declared that those were ineligible for patent protection, that is, that they lacked patentable subject matter, and the Federal Circuit ultimately affirmed that. So patent protection with respect to pieces of DNA like this, even if that DNA is synthetic and even if it has a particular function, I think that the patents would be subject to a pretty robust challenge. I think it's going to be tough. Yeah.
Great. Thanks.
Yeah. Thanks. So this is how the canonical CRISPR system works. Let me talk a little bit about profit and collaboration in the CRISPR area. I guess we're going to kind of move from questions regarding the patent eligibility or the patentability of this technology and more onto the licensing issues. What happens when someone has an already issued patent and they go about licensing it? What are some of the ethical, legal, and social issues that arise from that, right? So one of the ethical, legal, and social issues that arise from that is the conflict between research institutions and for-profit surrogate companies out there. One of the ways that this gets effectuated is that research institutions actually take an equity share in the startups that they have to develop this technology going forward. And that company in turn, right, has the right, generally speaking, to sublicense that technology to other smaller companies going forward. This may seem like best practices, but I actually think that this equity ownership, as opposed to a royalty arrangement, seems to present some conflicts for research institutions. Namely, it presents conflicts of coordination between the particular researcher that's involved and between other researchers who are working on similar technology. For taking an equity share in one company, it automatically seems like the technology that's being developed is a zero-sum game. That if Editas isn't doing well, if you're the Broad Institute, for example, nobody is doing well. To the extent that there's another researcher who is working for your institution that is doing something similar, you may not be as invested in their particular research. You may not want to take an equity share in their research. You may be less willing to allow them to sublicense their technology at particular royalty rates because it may seem to be cannibalizing from some of the equity that comes from the company from which you've exclusively licensed your patents to, right? That seems to be the kind of first particular issue there, right? This seems to be something that is occurring more frequently lately. There are a number of research institutions that are moving away from the royalty model, right, that are moving away from the running royalty model and are kind of going towards more of an equity basis. This particular graph from Nature Biotechnology, my apologies for not including the citation at the bottom of this slide, shows the kind of slow creep of cash-in equity over the years, right? Seems to have reached a high watermark in 2010 and 11 and again in 2014, while at the same time it seems like running royalties are kind of hovering at, roughly speaking, the same level, right? This is data, I think, from AUTM, right? It also seems like research institutions who are involved in this particular area seem to be detracting from interinstitutional research collaboration going forward. If it seems like the research institution involved cannot formalize a lot of the intellectual property licensing arrangements before the researchers collaborate, this may encourage research institutions to tell their employee scientists, you shouldn't be working with someone not only from the lab down the, that you shouldn't be working with your fellow colleagues at another research institution out there. We don't know what the intellectual property issues are going to be to the extent that there's some co-inventorship dispute between you two, and therefore one of the things that we want to do is we want to collect everything in-house. To the extent you want to collaborate, you should essentially only be doing it with people from within your same institution.
So just to clarify and elaborate on that, I mean, how are these issues different than what already exists in terms of these collaborations?
Yeah.
So does CRISPR, is it just the uncertainty over the IPRs now because of what's happened with the interferences, or are there?
So to be clear, I don't think that CRISPR has changed this. I think what CRISPR does is highlighting some of the problems that actually occur here, and this is actually particularly salient for the CRISPR technology itself, because there are such interinstitutional research conflicts with respect to CRISPR, right? There is one such conflict between Feng Zhang, who's at the Broad Institute, as I mentioned before, and another researcher by the name of Marafini at Rockefeller University in New York. They were actually co-authors on a science paper that tends to be credited to Feng Zhang that came out in 2013, and there seems to have been a major intellectual property dust-up between the Broad Institute and Rockefeller. There was a patent application filed in Marafini and Zhang's name. It's unclear whether the person who was doing this had authorization from the Rockefeller University, but it seems that there was a patent application filed in Marafini and Zhang's name to force the co-inventorship issue to be decided by the PTO going forward. There still seems to be some collaboration, I think, between Zhang and Marafini, but this certainly is going to lead to some tension going forward between the Broad Institute and the Rockefeller Institute. For that matter, as I think I'm trying to point out here, I think it's also going to lead to some tension between the Broad Institute and between any other institute to the extent they cannot a priori agree on some intellectual property licensing and sharing scheme. And I think that that's kind of problematic. I would rather live in a world where scientists felt I don't know, freer to engage in these interinstitutional collaborations without worrying about whether administrators are going to get upset because doing so would have violated the intellectual property assignment agreement that they signed on when they became employees, right?
Was there a relationship between Doudna and Zhongfeng as
yeah,
well?
right.
Adidas. I think I remember reading that in one of the
yeah,
articles.
right. So the story goes, and I actually don't know a lot of the detail of the story. I don't know if anyone does. So the story goes, Doudna and Zhang actually co-founded Editas together. I think, around the time that they were both coming out with their publications in science. But then it seems like Doudna left shortly thereafterwards. She recounts a little bit of the story in her recent book, co-authored with Stam Sternberg, called A Crack in Creation, and just says, something to the effect of, there just, there wasn't good chemistry. There just wasn't a good fit between what she was trying to do from a research perspective and what Editas was trying to do itself. That doesn't really tell us much about what actually happened. But again, that seems to, be a cross-institutional co-inventorship dispute, right? There also was a cross-institutional co-inventorship dispute, believe it or not, between Doudna and Charpentier. So for a number of reasons having to do with Sweden's lack of a Bayh-Dole Act, when Jennifer Doudna and Emmanuel Charpentier's application gets filed, University of California is the assignee of Jennifer Doudna's rights, but Emmanuel Charpentier held her own rights. So even though University of California, Berkeley was responsible for prosecuting the application, some evidence to suggest that Emmanuel Charpentier did not think that they were taking her interests into account, her individual interests into account as much. So this was leading to a tension of Emmanuel Charpentier assigning her interests in the patent application to other entities, kind of without the consent of University of California, Berkeley, or without discussing it with Jennifer Doudna, and between what University of California, Berkeley was also trying to do. That was kind of out there during the course of the interference proceeding, and then shortly after the interference proceeding concluded, there was a cross-licensing arrangement that was negotiated between kind of everyone who had taken a license on Emmanuel Charpentier's side and everyone who had taken a license on Jennifer Doudna's side. So that aspect of it seems to have been resolved, but it was an outstanding issue for roughly speaking 4 years. So it's
do you think that was
tough.
possible there because of the relationship between Doudna and Charpentier? I mean, was there that underlying collaborative, or I guess we're just speculating.
I mean, it's obviously tough for me to say. I don't know any of the parties personally.
No,
I mean,
Well, that's amazing that we talk about transactions costs, but if you're talking about all of these licenses that are being granted by both parties.
Oh yeah, it's
amazing
just
try to coordinate that.
It's just huge. Just simply, if you're a company who wants to work in the CRISPR space, just trying to figure out who has the license to your particular technology, who should you take a license from, is inordinately complicated, and it's not even like there is a right answer.
Right.
You're essentially betting on a horse race. And since the interference decision came out, some members of that horse race lost, right? And some of the people who have entertained large bets on the availability of licenses to this technology are nonetheless doing really important work. And if they end up, losing badly, they may be holding an empty bag, right? I think CRISPR Therapeutics, they're a publicly traded company. One of the things that they're working on is using CRISPR to cure sickle cell anemia. They have taken their license from Emmanuel Charpentier, right? And to the extent that Jennifer Doudna and Emmanuel Charpentier lose, CRISPR Therapeutics is going to have to start thinking about, do we need a license from the Broad Institute or Virginia Sheiksnies? And to the extent that we do, how much is that going to cost? For that matter, is the Broad going to negotiate with us on favorable terms? Maybe they won't.
I mean, it's interesting to compare the CRISPR story with the PCR story, which whatever your perspective, there's some people who think it should never been patented. We'll put that aside. We can have that separate discussion. But from a pure commercialization licensing perspective, I mean, I think that's PCR is viewed as a success story. And one of the thoughts I had comparing the two is that there just seem to be a lot more players and a lot more kind of For lack of a better term, I'll make it a little bit dramatic to make this webinar, kind of a little bit spicy, a lot more internecine type kind of
Yeah.
things. Conan Boyer, it seemed to be kind of a very traditional story. You had the researcher coming up with a breakthrough within maybe, one institution, maybe two institutions involved, and then they were able to move it forward, and you have the success story there. I mean, which I think, I don't want to simplify. It seems like here, maybe it's too early to say whether there's a success or not, but it just seems a whole lot messier, and it can only be that there are a lot more people involved trying to get this pioneer invention patented, and in that space. Maybe there's just the mentality has changed since in the last 37 years. So there's more pressure to commercialize. So it's kind of interesting to do that comparison. Even though I realize it's still early to say whether CRISPR has been successful or not.
Yeah, I mean, I think that there is some view that the personal riches that were experienced by, it's funny to think of riches as being experienced by someone in the passive tense, but that the personal riches that were gained by Cohen and Boyer, by Richard Axel for his calcium phosphate transfection method patent, but those were anomalies in the life science space, and that generally speaking, if you were a wet scientist, driving a Maserati is like not going to be in the cards, right? I think that view of the world is gone, right? We live in a world, not necessarily a terrible world to live in, but we live in a world of celebrity, rock star, super wealthy scientists. We live in a world where Stanford researcher Steve Quake sells his NIPT patent technology to another companies out there, and I think that netted his company $400 million or something like that. And he's someone who, obviously is an important player in my field, but is probably not a household name to everybody, right? So we do kind of live in that world right now. To kind of go off a little the beaten track here, right? There's this wonderful book by Margaret Atwood. It's the first in a three-part series called Oryx and Crake, and it's this kind of dystopian future, typical kind of Margaret Atwood story. And part of the dystopian future is this world in which there are, celebrity rock star scientists whose talks sell out stadiums and stuff like that. And don't know if we're there yet. Seems particularly ironic to say that this is occurring, especially in a world or especially in a country where, the vast majority of people, deny that there's climate change or don't believe that evolution is real or stuff like that. But I mean, I think we're kind of getting there, right? I mean, I think we're kind of getting there where the kind of Venn diagram between scientist and celebrity is starting to overlap more and more.
And maybe it's the success of PCR, however that's gauged, is what is kind of, if you want to call what's going on now a gold rush in the in the PCR, in the CRISPR space. That may be
that's possible. To use a really terrible analogy that also
that's what we specialize.
Yeah, it's what I specialize in. Use a really terrible analogy that is also anachronistic. You think about the PCR patenting story, right? Kerry Mullis, makes his riches because he licenses it out at like a dime to people who want to use the thermocycler. Everyone who's making a thermocycler, and then some people who otherwise ignore his request for licenses, he doesn't go after them, right? It's this policy of cheap licensing and non-enforcement at the same time. I would struggle to think that there is any research scientists out there who decided that they wanted to become rich by following Kerry Mullis's model. Rather, I think the research scientists out there who decided that they wanted to become rich probably followed the model of Richard Axel or Cohen and Boyer or something like that, where we're going to actually engage in this kind of robust licensing practice where we get people to glom onto our particular platform, and while they're not paying an arm and a leg, they're still paying something more than 5 cents or 10 cents or something like that. So here's where the horrible analogy kicks in. I don't think anyone grew up wanting to play guitar because they saw the Grateful Dead, or anyone wanted to be a rock star because they were Grateful Dead fans. The people who want to be rock stars listen to Led Zeppelin, right? I mean, so that's kind of what I think is kind of happening here, right?
Yeah,
You kind of look at Kerry Mullis with this policy of just like, who comes? If you want to tape off my soundboard, fine, right? That's the kind of Grateful Dead model, right? It has its adherence in the field, but it's not like propelling everyone to pick up a guitar and learn how to play Stairway.
Yeah, interesting.
Right? I mean, that's kind of what I think has happened now.
I have to unpack that and think. I mean, there is kind of an interesting
or
question.
not,
or not.
Yeah.
I mean, but
Sure.
it's very thought-provoking. I mean, when you're talking about celebrity, rock star scientists, another name from Stanford that came to mind was Carl Djerassi. Dr. Carl Djerassi invented the pill, or did the background work for the pill in the 1960s.
Oh, okay, wow.
I mean, he had a kind of an interesting personality as well. I don't know, I mean, whether it was motivated by the kind of things that you're talking about, but that's sort of another model, which is maybe more of a laid-back 50s, 60s style model than kind of a post-80s. But I'm just throwing that out there.
Yeah.
I don't know if that's an analogy or not. It's just a kind of another historical data point of a scientist who really had a big impact.
Yeah. I mean,
and not just simply in the lab, but just really kind of became a personality.
Yeah, definitely. I mean, that's so, that we have this, wonderful diversity of models today, and one aspect of those models include people who are really after it for glory, riches, and fame, and that's not necessarily bad,
Interesting.
Anyway, wonderful.
Anyway, continue.
Sure. Yeah. No.
Yeah.
Please. Yeah. Lovely. So, in any event, all right. So, kind of enough about some of the interinstitutional collaboration issues. I want to kind of hone in a little bit on some of the difficulty with respect to for-profit surrogate licensors. So George Contreras and I published a perspective in Science earlier this year talking about the complicated web of licensing arrangements that happen to have occurred from the CRISPR patents and the CRISPR patent application out there. A lot of this is mediated because the research institutions involved, rather than being responsible for rather than being responsible for sub-licensing their own technology going forward to individual companies working on a specific gene or working on a specific application, actually farm out their sub-licensing practice to these for-profit surrogate companies. These for-profit surrogate companies take an exclusive license, almost wholesale, to the intellectual property with respect to that technology that's owned by the research institution. And then these for-profit surrogates then end up sub-licensing the technologies to certain companies out there who are engaged in particular applications. This is a pretty graphic model that George Contreras and his research associate have developed, showing exactly how the surrogate licensing model works. On the left, you obviously see the institutions and patent holders that are involved with respect to the particular patent applications here. In the middle are the surrogates, and on the right are the kind of sub-licensees. The surrogates are doling out sub-licensees to the licensees that you see on the right here, right? Part of the problem with surrogate licensing is this kind of conflict between equity ownership and development and access that I talked about earlier. Another big problem is a competitive one. Another big problem is competition. The surrogates are not in the sole business of granting exclusive licenses to develop certain applications of the technology. Rather, the surrogates are also in the business of developing the technology themselves. So by doing so, they are direct competitors with the companies that they are doling out sub-licenses to. This is particularly problematic because this means that the surrogates, as a competitor in the marketplace, essentially have the right to ask others or to tell others whether or not they are going to be allowed to obtain a license and on what terms to develop a specific application of the technology, even if that's one that the surrogate themselves are not necessarily doing. That hasn't absolutely happened thus far. I think that is important to point out. We haven't heard of stories of companies going to these surrogates and saying, we want to do something completely different from what you are doing, and the surrogates saying no, but we have had a number of instances of the surrogates granting sub-licenses that are far too broad to be practiced in full by any of the sub-licensees that they are granted to. I think the salient example here is probably the one at the very top between Editas and Juno Therapeutics. Juno Therapeutics, and this somewhat of a dated reference at this point because they're slowly losing the CAR T race here, but Juno Therapeutics was responsible for developing or for pushing or attempting to push through clinical trials a particular version of gene therapy technology called CAR T. In a very brief description of this, CAR T stands for chimeric antigen receptor T cell. What the therapy does is extract T cells from the white blood cells in your body, modifies them to attack a particular cancerous cell, to attack a particular antigen on a cancerous B cell, for example, and then injecting these modified T cells back into your blood as a form of therapy. There's ways to do this with CRISPR. There's ways to do this without CRISPR. Editas and Juno have arranged among themselves a cross-licensing agreement such that Juno is the only company that has a license from Editas to use CAR T technology, use CRISPR CAR T technology, I should say, independent of the particular target that is sought to be effectuated, right? If you want to develop CAR T technology for one particular form of B-cell lymphoma, that's Juno. If you want to develop CAR T technology for a different form of B-cell lymphoma, that's still Juno. And it's going to be Juno for all of the applications of this particular intersection between CRISPR and CAR T, even if that is a greater number of things than Juno is actually able to research themselves. They are locking other competitors out of the market who otherwise may want to develop this technology, even if it's not something that Juno is doing themselves.
One thing that comes to mind when I see this chart, it's really fascinating, is this tension or this difference between general purpose technology and specialized technology, or the bespoke technology, whatever you want to call it. And that sort of comes up in the software space. And so I'm trying to run through my head sort of analogies in software, right? And the CRISPR technology has, it's word processing for genes in some level.
Yeah,
right?
yeah.
And so I think there's more than kind of a superficial, it's not as great as your Led Zeppelin analogy, but there is sort of at least a superficial analogy here with what happened in software, right? So I wondered if that seems, shoot that idea down.
Yeah, no.
Whatever.
So I actually I think that is readily applicable, and this is actually something that George Contreras and I talk about in our science paper. The way that we view CRISPR is not so much as a research tool as it is a technology platform. It's the platform on which a lot of future applications of technology are hopefully going to be developed. So it's perhaps appropriate to think of CRISPR in the same sense as, say, a Wi-Fi standard or the kind of Bluetooth standard essential patents out there, right? This is the kind of fundamental aspect. This is the platform of technology.
It wouldn't be like an operating system. I think of software analogies, operating system with apps, or I guess, again, analogies can do whatever you want them to do.
Yeah, sure. Maybe an operating system would be good. Although, I'm going to just completely confess is that I don't know enough about the guts of how operating systems work. I know kind of vaguely that it's a processing kernel, and that's essentially where my knowledge ends. Not necessarily my field. But yeah, it definitely is a platform technology in which a lot of applications by others, probably not known at the time the platform is developed, are going to propagate the technology going forward. Yeah.
And the only reason I hesitate a little bit with the Wi-Fi technology is that there's a little bit of a chicken and egg issue with that. As to whether it's the handsets that were developed in light of the Wi-Fi, or it was the other way around. So
sure.
Yeah, it's not clear what's the platform, what's the
that's fair. If you want to draw that out a little bit, right?
Let's go back to Led Zeppelin.
Yeah, sure. No. But look, if you want to draw that out a little bit, right? The kind of reason that the reason that CRISPR is important is because we're faced, I hate to be too romantic, capital R, about this, but we're faced with the mortal handset that we have, right? Essentially we are operating under the genome that we have, not necessarily the genome that we want, hence the desire to edit it in the first instance. So What's really the platform technology, the human genome or CRISPR? I don't know. Depends upon your philosophical perspective of that stuff.
Right.
But I mean, yeah, that's, CRISPR is still probably best considered to be a platform technology on which future applications are going to be developed.
Interesting. I think that's reflected here, or at least one way to read, these things. So it's great. Thank you.
Yeah, well, wonderful. So let me just make my way to my last point, seeing we have 10 minutes left, and then kind of happy to take any questions.
Sure.
I think a lot of the things, or the kind of portrait that I have painted of the CRISPR licensing so far has been generally bad news. Look at these effects that they're going to have in the interinstitutional research collaboration platform. Look at the conflicts of interest that are going to arise when surrogate licensors happen to be taking control over who gets a sublicense to the technology. But I don't want to suggest that it's all bad. Right? One of the really important aspects of this technology that we can still use CRISPR patents for is as a function as an ethical gatekeeper. This is something that Christy Guarini and I and Maggie Carnuti and Chris Scott have written about in an early 2017 Nature Biotech paper, which we describe as the rise of the ethical license. Right. So one of the advantages, and here's a little snippet from the paper. One of the advantages of patents is that by prohibiting uses the patent holder deems unethical, the patent holder can determine who gets the license of the technology and on what terms. And those terms don't just have to be financial terms. Those terms can be ethical terms as well. They could be requirements, for example, that the person who's obtaining a sublicense to engage in this technology is only going to use it subject to rigorous scientific and ethical controls going forward. This isn't just kind of daydreaming. This has actually been proposed by a researcher at MIT by the name of Kevin Esvelt. Kevin Esvelt is one of the, I guess the best way to describe this is leading thinkers on this one particular aspect of CRISPR technology called gene drives. Gene drives is a variant of CRISPR where the particular gene that you would like to insert into the genome is structured in such a way, the single guide RNA is structured in such a way as to drive only a single variant of that gene through a population every generation going forward. So let's say, for example, that you have a copy of alcohol dehydrogenase 1, right? And you want to make sure that everyone for future generations only has 2 copies of alcohol dehydrogenase 2, right? Well, in theory, you could use gene drive to do that. Gene drive would essentially find every instance of alcohol dehydrogenase 1 and replace it with alcohol dehydrogenase 2. You could think of it, and again, since we're kind of in the market for bad analogies in this webinar right now, you could think of it as a kind of search and a perpetual search and replace function for a particular gene.
And you're talking about that within an individual organism, is that correct?
Yes, but to the extent that it modifies the germline, it gets passed on.
That's my next question is how do you go from that to the population? But your idea is that over time, it will populate that way.
Yeah, right. And so this has been
virus or whatnot.
Right. So this has been demonstrated as some limited effect in mosquitoes, for example. Hence this graphic of a mosquito with some kind of drive being put into it. I would love to work into the art department for a science journal. That seems like a really awesome job. Anyway. So this has been demonstrated as some limited effect in mosquitoes as a way of controlling. And also I believe in the white-footed mouse in Martha's Vineyard as a way of controlling Lyme disease. So that's what its proposals are for. The problem is that because it's perpetual, the problem is because it's ongoing, and the problem that because it's going to modify the germline, it gets passed on to future generations. This raises some ethical concerns. So one of the things proposed with respect to gene drive by Kevin Esvelt is using patents as a means to essentially prevent others from using the gene drive technology at all for research or for commercial purposes unless they subject themselves to rigorous scientific or ethical controls.
And would that be something, we would have to amend or create a new agency or alter the FDA, or what would that entail? There's a lot of speculation here.
So a lot of it
presumably the FDA would not as it currently is structured, would not be able to deal with something
So,
like that.
yeah, so a lot of it is going to depend on what the organism is that is being modified,
Yeah.
what the gene is that is being used to modify the organism with, and lastly, whether that organism is intended for use as food. No one is eating mosquitoes.
Yeah, right.
Don't think the FDA really has a lot to say
Yeah.
about this. This is more stuff for the EPA, maybe
Okay.
to a
Yeah,
certain extent, the Department of Agriculture. Yeah, that's
Or a
kind
new
of,
agency, yeah.
yeah, or for
Yeah.
some matter, state wildlife, fish, and game agencies with respect to the proposal to use CRISPR gene drives on white-footed mouse and Martha's Vineyard, or using these kind of malaria-proof mosquitoes in Key West in Florida.
And for disease therapies, I mean, for example, cancer, I assume, to the extent that we can figure out sort of the genetic, or we want to control kind of the uncontrolled growth that occurs and the mutations that occur. I mean, it's not possible yet, but within the individual body, would that be possible to make these alterations? To fight
Yeah,
off
I
cancer
mean, it's
cells,
possible.
or?
So, I mean, there is some cancer
Yeah.
CRISPR screen work going on to determine exactly which genes, develop a particular tumor or not.
Right.
So, I mean, yeah, it's possible.
Let me just ask the kind of naive consumer question. If you already have cancer, would it be possible to use CRISPR to alter the existing cancer cells?
Yeah, I suppose it's possible. I mean, to the extent that there is a way to target only cancer cells, and if you can remove cancer cells' ability to divide, if you can essentially arrest cell division by removing some of the genes necessary to do it, then in theory, you've killed off the tumor at that point. And that is extremely theoretical. I don't know if anyone's attempting to do that right now, but I mean, I guess it's possible. Again, yet another potential application for CRISPR. Yeah.
And what do you think about the ethical implications of something like that? Is that something that would be readily handled under current regulations?
Yes,
with
I mean,
the FDA?
that
would be a drug or some sort of,
I mean, that is something that decidedly would be a drug or a biologic in the same way that those things are regulated.
I guess
now.
It's like a biologic, yeah.
So it's actually going to depend. So there's a lot of oligonucleotide therapy out there right now that is actually regulated as a drug. So Sarepta's Exondys is actually regulated as a drug and not a biologic.
And what is that exactly?
That is an RNA interference therapy. It's again the short snippet of RNA that is meant to I guess in this particular case, to produce a protein that otherwise is not being produced previously.
I see. Interesting.
Yeah. So I see we only have 5 minutes left. I think it makes sense for me to stop here and take any questions that anyone in the audience has. Trying to take a look at that.
Yeah, I don't know if we have a list of participants, or maybe we do. Yeah. I think we've got we had some good questions before. Let me just kind of try to move to a conclusion here for this.
Sure.
Right. I mean, I think people are found it very informative. So thank you, first of all.
Oh, please.
For this time. So I think it's really helpful.
Thank you.
When you're looking at the ethical issues, I mean, do you think a lot of these ethical issues could be dealt with through new regulations or rethinking? Or do you think there are some insurmountable issues that we're just going to have to rethink at the societal level or cultural level?
I don't think there's anything insurmountable, especially because there hasn't really been anything horrible yet.
Okay.
I mean, even the kind of worst case scenario of these gene drives kind of get out of control, and everyone, for example, has a copy of alcohol dehydrogenase 2 as opposed to 1.
Mm-hmm.
I mean, that's bad. I mean, it's not nuclear war, right? So I mean, I don't think that there's any kind of issues that are totally insurmountable that would make it such that any hopes of regulation would be impossible. The laws we have in the books today, assuming they're enforced sensibly and evenly, are not bad. They, by and large, tend to do their job.
Right.
So I mean, I think the science is relatively well positioned within the legal framework to advance without having to worry about some regulatory agency coming down on researchers, right? Time will tell, obviously, to see whether the technology delivers on its promises or spirals out of control. right? That's something that we're going to have to just kind of wait and see.
Yeah. No, this is a great presentation. Thank you. And so we'll have the video up on our YouTube channel in about a week or so. And I think Professor Sherkow has a number of papers on this, and we can make those available.
Sure,
to
please
the
do.
YouTube placement as well. And just leave it to you to have the final word if you wanted to, leave us with words of optimism or words of dread, as I like to put it. So.
Yeah. So, obviously one of the most important pieces of technology been developed, I don't know, just kind of period in the past 50 or 60 years. I'm really excited to see what the future is going to bring.
Okay. Well, thank you again. It was great to have you, and welcome.
Thank you. Thank you very much
to the series. So
Thanks.
thank you again.